All right, thank you, Guy. I'm going to skip through the first part here since we're a little limited on time. Basically, I just did some RNA-seq and proteomics looking at our glial overexpression model. I'd be more than happy to talk to you about this later, but I'll skip to the work that's the title of my talk. So, as Dr. Ryder mentioned yesterday, we used fruit flies to try to look at the genes in the Dup15q region. So I'm interested in what other genes, other than UB3A, could be driving some of these phenotypes. So I'm sure we all know that there's a lot of genes located within this region. One of those is UB3A. Another gene located in that breakpoint 2-3 region is HERC2. So being located in the 2-3 region, just about everybody has extra copies of HERC2 and UB3A. And so just a quick background search through the literature of what's known on HERC2, you actually find that HERC2 mutations result in Angelman syndrome-like phenotype. So recently, some patients were identified that had a deletion that fused the first 56 exons of HERC2 with the neighboring gene OCA2, and this was expressed under the HERC2 promoter. And it lacked a couple important domains from HERC2, so it didn't actually have the RLD2 and RLD3 domain. And these individuals had uncoordinated movements, growth delay, a lot of symptoms that are similar to Angelman syndrome. Another group was found to have this proline 594 leucine mutation, and again they had developmental delay, this unstable gait phenotype, and 4 out of 15 of those individuals actually had seizures. And a second group also found a family with this proline 594 leucine mutation, again with motor delays and ASD-like phenotypes. So this is an indicator that another gene located within this 15q11.2 to q13.1 region, when mutated, will be driving some cognitive phenotypes. And early work out of Martin Scheffner's lab actually found that HERC2 interacts directly with UB3A. So they did a yeast-2 hybrid screen using E6AP or UB3A as the bait. And when they went through their whole cDNA library, they only found 5 cDNAs that actually were interacting with UB3A, and 3 of those were different parts of the HERC2 protein. They also did a fused UB3A to GST and did pull downs, and whenever they did mass spec on that high molecular weight band, they actually found that in that band was HERC2. So this is again indicating that HERC2 and UB3A are physically interacting. A second group, Peter Howley, they expressed different fragments of HERC2 with the HA tag and did pull downs. And whenever they expressed the fragment of HERC2 that contains this RLD2 domain, they actually found it binding with E6AP or UB3A. So again, just another lab confirmed this interaction with UB3A and HERC2. And Dr. Schafter's group interestingly showed that HERC2 actually increases the ubiquitin ligase function of UB3A. So this is just an in vitro ubiquitination assay where they stick UB3A in a tube with all the necessary subunits. Over time, UB3A actually auto-ubiquitinates itself, so we start to see these high molecular weight smears here. Whenever they incubated UB3A with a specific domain of HERC2 called RLD2, that actually increased the rate of this auto-ubiquitination. And whenever they took a different part of HERC2 protein, the RLD3 domain, you didn't see this increase in auto-ubiquitination. They also found that not only did RLD2 domain increase auto-ubiquitination of UB3A, but also ubiquitination of a target protein, ring 1B. Now, so moving into Drosophila, fruit flies have a single homolog to HERC2. It's also called HERC2. You can see a lot of the domains are actually shared between human and fly HERC2, in particular the RLD2 domain, so that's the domain that's shown to bind to UB3A. So flies also have that domain conserved. And lucky for me, there was a fly line that had a UAS element that landed right upstream of the HERC2 start site, so I can actually use the GAL4 UAS system to overexpress HERC2 at about 5 to 7-fold, depending on the GAL4 driver that I use. And so this work that I'm talking to you about today was done in neurons, not glial cells, so I moved back to neurons. And the first set of experiments I did was a simple behavior assay, a climbing assay. So it was known that if you overexpress just UB3A, so this green bar, flies have a climbing defect. So it takes them longer to climb up in the tubes. Whenever you overexpress just HERC2, so the blue line, we don't really see an increased climbing defects. But when I overexpress both UB3A and HERC2 at the same time, so this red bar, it actually exacerbates that climbing defect. So it takes them even longer than just UB3A overexpression alone to climb up the tubes. So this was just kind of the first piece of evidence to me that there might be something going on my flies when I overexpress UB3A and HERC2 at the same time that I'm not seeing with just UB3A alone. So this has been hinted at a little bit, that there's this kind of co-activation function of UB3A, so it can act to actually increase the transcription of some proteins. In flies, one of these proteins is called punch-RB. So Dr. Ryder's lab showed a while ago that UB3A acts as a transcriptional coactivator, so when UB3A levels are elevated, we see this upregulation of the punch transcript, and that's shown here. So when I overexpress UB3A neurons, I see about an 8-fold elevation of this punch-RB transcript. Whenever I overexpress just HERC2, I don't see this elevated punch transcript, but when I overexpress both HERC2 and UB3A at the same time, the coactivation function of UB3A is actually reduced. So it goes from about 7 or 8-fold down to about 3. And this is not simply due to reduced amounts of UB3A transcript. Overexpression of UB3A and HERC2 at the same time has about a similar overexpression of UB3A or compared to UB3A alone. So next I wanted to actually look at the protein level of the UB3A protein. So here's my flies that when I overexpress just UB3A neurons and take off their heads and do a Western blot, we actually see an elevation of UB3A protein, which is good. If I overexpress just HERC2, I actually see reduced levels of UB3A protein compared to controls. And when I overexpress HERC2 and UB3A at the same time, we actually don't see quite as high elevation of UB3A levels. And all this is just quantified here. So if you compare the green bar with the red bar, adding HERC2 to UB3A overexpression actually goes down. And then the blue bar, which is just overexpression of HERC2, we see reduced compared to controls alone. And this is exactly what you would predict based on Martin Scheffner's data with HERC2 increasing the ubiquitin ligase activity of UB3A. You would actually see that downregulation, which is what I'm seeing in the flies. So next I did a pretty big proteomics run to try to find what proteins are changing due to overexpression of either DUB3A alone, HERC2 alone, or HERC2 plus UB3A. So we did CMT labeling and trypsin digest and then ran it on the mass spec. First pass analysis of this, we identified about 5,000 total proteins in the fly head. So that's pretty good coverage, we think, considering the whole fly genome is about 14,000 proteins. One thing I'd like to note is that there's a large number of proteins shared between overexpression of UB3A alone or HERC2 alone or HERC2 plus UB3A. And I think also an important thing to note is there's actually a lot more proteins that are changing levels when we overexpress HERC2 compared to just UB3A. So, so I tried to dig into to try to understand what some of these proteins are that are actually changing. And for this, I use protein set enrichment analysis. So you might be familiar with like a gene set enrichment analysis for RNA-seq data. This is a similar tool developed by the Yates lab, who are proteomics leaders in the field. And whenever I ran this analysis on my flies that were overexpressing both HERC2 and UB3A, I actually found found enrichment for things related to RNA, such as mRNA processing, mRNA binding, RNA metabolism, among a lot of other things. And one of those proteins was actually FMR1. So you can see here overexpression of just UB3A alone. We don't really see any upregulation of FMR1, but when we overexpress HERC2, FMR levels go up, and we overexpress both UB3A and HERC2 at the same time, these levels get even higher. And that's something that I want to try to follow up into more now. There's really good antibodies for FMR1 in the fly, so that's something I'm going to try to look into a little bit more. And so since we knew that there's kind of this idea that HERC2 is upping the activity of UB3A, I wanted to try to look at what proteins are starting to be downregulated as we have both HERC2 and UB3A elevated at the same time. So I dug through this data set, and what I hope you can appreciate here is there's a lot more things that are black and red in this column, which is the overexpression of HERC2 and UB3 at the same time, compared to these other two. So I pulled out significantly differentially downregulated proteins only when HERC2 and UB3 go up. And you can see that some of the proteins start to be downregulated when you overexpress UB3 or HERC2, but they all start to move down as we overexpress both. So I think these handful of proteins might be specific targets or specific proteins that are only downregulated when both HERC2 and UB3 proteins are elevated. And some of us last year were lucky enough to hear Dr. Scheffner talk about this interaction between HERC2 and UB3, and he kind of described the interaction as HERC2 acting as like an allosteric modulator of UB3. So whenever you have HERC2 around, it might be changing possibly substrate specificity and definitely activity of UB3. So this list of proteins might be some of those specific target proteins that he was kind of hinting at. And these proteins are involved in things like protein transport and the Golgi and vesicle-mediated transport. And these are some of the same gene ontology terms that Peter Howley's group identified from their HERC2 binding proteins. So I hope what you can take away from this is that whenever we overexpress HERC2 at the same time as we're overexpressing UB3, we might actually start to see, we'll see things that aren't the same as overexpression of just UB3 alone. And that kind of speaks to the fact that Dup15q isn't UB3 overexpression syndrome, it's Dup15q syndrome, so there's a lot of genes in this region, and we can't forget about them. I think UB3 is definitely a clear player, but HERC2 and I think a lot of other genes in the region are going to start to play a larger role. And with that, I'd like to thank my mentor, Dr. Reiter, some people in the cores, Dr. Daniel Johnson and David Chaknia-Shavelli, who helped with the proteomics. We also had some proteomics help from Daniel Lopez-Farr at Thermo Fisher, members of my thesis committee, and a big thanks to the Dup15q Alliance for supporting me.